Dear students, today I talk about research methodology and biostatistics, a series of lectures which is a precursor for any nutrition or dietetics student, for the matter of fact for any research student to conduct their research. It starts with hypothesizing your problem and making a sample size and sampling and identifying the correct design, and finally using appropriate statistical methodology to infer your results. So therefore, as a series we start with today sample size and design of studies and how to calculate the sample size, what are the different sampling methods that have been used to pick your sample, and what are the different designs of studies are there, so that you can do different designs for different types of your problems. Why you need a biostatistics or research methodology? Because no two individuals in this universe are same. There exists a variation. Patients vary, physicians vary, nurses vary, hospitals vary, measurements vary, disease states vary, immune response varies, drug adherence varies, diet consumption varies between individuals. Therefore, to capture that variation, we have to use the sample and sample size and research methodology as a matter of extracting the variation. The principles of good experiment is based on 3 points. One is replication, randomization, local control. Replication is nothing but repeating the experiment as it is, other words, is called a sample size. Randomization, to minimize the bias, we randomly allocate the subjects to different groups. Local control is nothing but making heterogeneity into homogeneity. Anatomy of research, basically we have to follow 7 steps to achieve your outcome. One is you have to define the problem. 2, you have to specify the objectives. 3, you have to select the design or type of study. 4, select study population. 5, calculate appropriate sample size. 6, analyze the data. 7, determine the conclusions. If coming one by one, you have to define a problem. That means that you have to clearly state the problem that should permit clear description of overall goals and specific aims. Therefore, we have to have an hypothesis to prove or disprove of your research problem. What is an hypothesis? A well-formulated hypothesis will be both quantifiable and testable. That is, involve measurable quantities or refer to items that may be assigned to mutually exclusive categories. So, as an example, if you want to put an hypothesis, the hypothesis would be like this. For example, for males over 40 suffering from chronic hypertension, a 100 milligrams daily dose of this new drug lowers diastolic blood pressure on an average 10 mm per Hg. Therefore, it has involved many things. One is you have to identify the population from this. The population is male subjects who has the age of 40 years and who are suffering with hypertension is our population. From that population, you have to pick the sample. Therefore, you have to specify the objective to prove your hypothesis. The objectives of the study should be clearly stated, and there should be a description on how the objectives are related to the design of the study. Both primary and secondary objectives should be included. So, the third point comes, and which we are going to discuss now in detail, select the design or type of study. What do you mean by design? The design of the study is the procedure and method predetermined by an investigator to be adhered to in conducting a research project. The methods used to obtain valid data to answer a research question. Therefore, what are the different types of designs you have? There are observational studies, there are experimental studies. What is an observational study? An observational study collects data from an existing situation. The data collection does not intentionally interfere with the running of the system. An experiment is a study in which an investigator deliberately sets one or more factors to a specific level. So, what are the different observational studies that are available? One is a correlational study, case report and case report series study, cross-sectional survey, case control study, cohort study. In experimental, randomized control trials or community trials. We will go one by one about the observational studies. So, one of the important observational study is the cross-sectional design. Whenever you want to estimate the current scenario of the population or in terms of the prevalences, the cross-sectional design is most commonly used. It includes surveys, laboratory experiments and studies basically to estimate or describe the prevalences of the diseases at the present scenario or the current point. So, a research objective is often used with a cross-sectional study, as these observation studies are often descriptive in nature. Case control studies where investigators are seeking information on past behaviors. For example, two groups of subjects are selected on the basis of whether they have or do not have same condition of interest or some condition of interest. One group have a condition of interest, that is cases, and another group will not have the condition, that is control. Individuals in both the groups are then questioned about the past exposure to possible risk. A reasonable conclusion will be drawn between the condition and exposure. So, how to conduct a case control study is being depicted in the picture. So, what are the advantages of case control design? Valuable for studying rare condition, short duration, relatively inexpensive, relatively smaller samples needed. The outcome is always in an odds ratio. So, what are the disadvantages? Limited to one outcome variable. There is a potential bias from selection of cases and controls, does not establish sequences of events, and potential survivor bias, and also does not yield absolute risk estimates. Therefore, the case control studies are useful to assess the cause and effect of the past history of the subjects. Another design, which is a cohort design, is also called as a prospective or longitudinal design, where whenever you want to see the dynamic changes over a period of time, we use this cohort study, which is also called as a prospective cohort study. Prospective cohort study, where selecting a sample of the population, measure predictive variables or risk factor present or absent, follow up the cohort, measure outcome variables, this is whether the condition is present or absent. So, cohort studies, the advantages being, so whenever we are using a cohort studies, that is basically to identify or to extract the dynamic changes over a period of time, where you can take different types of follow-ups in different time intervals and assess what is happening over a period of time. The advantages being logical process of exposure to outcome, establishment of absolute risk, or we can call as a incidence sometime. Exposure can be measured without bias and can assess multiple outcomes also. So, there are many factors where, for example, smoking and cancer, adolescent growth and development, these are the things where you can use cohort studies. The disadvantages being very large numbers will be needed for an uncommon outcome, even larger numbers are needed for rare disease, expensive because of the resources needed to follow people over a long time, results not available for a long time, usually restricted study of exposures which are measured at the start of the study, and increasing knowledge may reveal far more important. Randomized control trials, where similar groups of individuals from the same source of population are allocated at random to receive or not to receive an intervention, then observed for occurrence of the outcome. So, from the group of subjects or patients, a group will be randomly allocated to treatment, another group is randomly allocated to control and follow up for some time, the prescribed time, and at the end you compare the results. This trial is more robust because it is controlled, all the situations have been controlled. The advantages of the randomized control trials being comparability due to randomization and same effect of known and unknown confounders gets eliminated. Experiments provide strong evidence of cause and effect, allows standardization of the eligibility criteria, maneuver and outcome assessment, allows use of statistical methods with few inbuilt assumptions. The disadvantages being may be expensive in terms of time, money and people, Many research questions are not suitable due to ethics, likely cooperation or rarity of outcome. To a greater or lesser extent, randomized control trials tends to be an artificial situation. Now we are going to talk about two sequence, two period crossover design. Initially, the subjects were randomized and allocated to test group and the reference group, and after the conduct of the experiment for a week or 2 weeks or 3 weeks set by the scientist, and we take the readings of the test group and the reference group, and we will wait for some time of the washout where the effect of the drug is or the therapy or the intervention is removed. Again we will swap the test to reference, reference to test, and we again conduct the experiment for a week or 2 week or 3 week suggested by the scientist, and finally we measure the values and estimate the results. The advantage of this is each subject has an advantage of having both the experiment and control groups, and 1/4 of the parallel group sample size is enough for this kind of a crossover design. Different from parallel group design, have to see period effect, and also period versus treatment interaction also we have to see. If at all we want to go and estimate what the relative strength of various study designs, the clinical trial is the most stronger design, followed by cohort study, followed by the case control study, followed by the cross-sectional study, followed by the case series study, and finally the weakest design is being case report study. And if at all you want to see the suitability of the study design where appropriate designs are being used, if at all your issue is diagnosis, a cross-sectional study is appropriate. If at all your issue is therapy, the randomized control trial is appropriate. If at all yours is a prognosis kind of a problem, you have to use a prospective cohort. If at all yours is a cause and effect, then go for a cohort or case control study. If at all yours is a descriptive study, you should go for a case series or cross-sectional studies. However, more than one study design can be used to answer any given question of causal association. Now we are coming to selecting the study population. Selecting study population is also very important. You have to see the appropriate population to get your appropriate sample, otherwise the results which you are going to depict are going to be wrong. Finally, we are coming to calculation of appropriate sample size and picking up the appropriate sample. What is the importance of estimating of a sample size? One of the common mistakes in research is failure to estimate an appropriate sample size before embarking on a research project. If the sample size is too small, even the best study cannot detect an important effect, and this can contribute to further confusion surrounding a topic. The process of estimating sample size can be technically complex, and of course a statistician intervention is essential in this process. So why is sample size calculation is important? Because of three reasons. One is economic reasons, second one is ethical reasons, third one is scientific reasons. Economic reasons in the sense, as we are taking only sample, not we are taking the entire population into consideration, you are likely to save your resources, time and logistics. The very sanctity of the sampling is to reduce or minimize the logistics. Ethical reasons. The ethical reasons in the sense, if at all you take a less sample size which is required than the one which you are planning, then if at all you are taking animals and sacrifice and got the results, then it would not prove your hypothesis, then you are doing ethically wrong. Scientific reasons, as there are sound scientific formulas are available to calculate the sample size, you should go ahead with the formulas and use those formulas and calculate appropriate sample size. Before you calculating the sample size, you also should know about how to pick a sample or a sampling. What are the principal steps that are being used or that are in the sample survey? So there are different steps to pick a sample or to conduct a sample survey. One is you have to write down the objectives of the study. A lucid statement about the objectives you have to write down and keep it ready for your study. Population to be sampled. You have to identify the right population for collecting the sample and use that sample only into your research. Data to be collected. You have to, before going for a survey or research, you have to identify what type of data, what are the variables you require to include into your research, you should identify before. Degrees of precision, I mean to say degrees of precision is nothing but since you are taking sample, you are not taking the entire population into consideration, you are likely to commit some error. That error you have to identify whether it is a 5% precision or 1% precision or 0.1% precision. The method of measurements. So, you also have to priorly define what are the different instruments you are using to get your data or to estimate your results. The frame, you have to make a frame of all these above 5 points and ready for taking or collecting the data. Now comes to selection of the sample, how to get the sample. There are different methods available, different sampling procedures are available to pick the sample, which we will discuss in the due course. The pretest, before going for a real survey, you have to do your pretest whether all the subjects which you are planning or all the questions which you are asking are getting appropriate answer or not. Organization of the fieldwork, it depends upon the managerial skills of the individual scientist or individual researcher to get the sample. Finally, you have to summarize after you collect the data, your results and analyze the data, and finally you have to write down the inference of your thesis. So, what is the advantage of this? The information at present whatever you are getting will be used for the future surveys, taking your results as the baseline of the research which they are going to conduct. So, what is a sampling? What, knowingly or unknowingly you are doing sampling every day. In olden days, our grandmother used to see a grain of rice and see whether the rice is cooked or not. That is also sampling. Therefore, now we are using some statistical and mathematical formulas to pick the sample, which we call as a sampling. So, a sample is a finite part of the total population. So, we pick the sample, identifying the population. From that, based on the sample size which you have calculated, you pick those many samples and analyze the data and interpret the results. So, what are the different types of sampling procedures that are available? There are non-random sampling, there is a probability or random sampling. The non-random sampling basically is a convenient sampling. For example, you take a snowball sampling, which is also non-random sampling, but is very important. This is typically used in qualitative research when members of a population are difficult to locate or for a covered subpopulations or non-cooperative groups. So, recruit one respondent who identifies others, so on and so forth, go on picking the sample, primarily used for exploratory purpose of, and also it is called the non-random sampling. There is also a convenient sampling, there is also a purposive sampling, there is also a quota sampling, which are not random but all are non-random samplings. In the random sampling procedures which we mostly use for scientific inquiry, there are 4 important random techniques. One is simple random sampling, second one is stratified random sampling, third one is systematic random sampling, the fourth one is cluster random sampling. So, starting with a simple random sampling, it is a method of selecting some small or sample units from the population where everyone has got an important or everyone has got an equal chance of being included in the sample. So, generally we have knowingly or unknowingly we have been doing since our childhood. Suppose we want to pick one person, we used to put all the names in a box and pick that person name. That is also nothing but a simple random sampling. So, simple random sampling has a disadvantage of getting the sample from one direction or one place. To overcome that, we use the random sampling called stratified random sampling. Stratified random sampling. In stratified random sampling, the population of n units or the total population which we are considering for our research is first divided into subpopulations or stratums, which we call respectively. These subpopulations or stratums are non-overlapping. So, we know the characteristic of each substratum and together they comprise the whole of the population. So, if we add all these stratums, the total population will be formed. Therefore, from each stratum again we pick some sample randomly, and from all the stratums whatever the random sampling you picked, we add them and we make a final sample, so that whatever the sampling or whatever the subjects we have picked is the true representation of the total population. Therefore, the heterogeneity is minimized or reduced when compared to simple random sampling. Therefore, the stratified random sampling has an advantage over simple random sampling to minimize the bias or heterogeneity. Now, we will talk about systematic sampling. Systematic sampling eliminates the need for complete up-to-date population but requires a reasonably accurate plan showing all households on an orderly layout, which makes it possible to go systematically through the whole site. For example, if at all we have the systematically numbered subject, say 5000 subjects, from 5000 you want to pick 100, then 5000 by 100 is 50. So, every 50th subject will form your sampling unit. Therefore, it starts at 50th and ends at 4950, so that all the 100 subjects is taken from the bottom to top, starting from the 50th subject ending with 4950. This systematic random sampling is having an advantage over stratified sampling over simple random sampling, as it minimizes the heterogeneity or variation in the sampling procedure. There is another sampling technique called cluster sampling. Here, the sampling unit consists of a group or a cluster of smaller units that we have called elements or subunits. For example, to assess the prevalence of Gaita in school children of a state, we may need to have a list of schools in various PHC areas or urban blocks or census towns. Each school constitutes a primary sampling unit. The children in each primary unit are the final sampling unit in this prevalence study. This cluster sampling is quick to get the sampling, but has got a disadvantage, the possibility of all the contiguous sample come in one place. To overcome this, generally whenever we are using a cluster sampling, we use the design effect. In general, we use the design effect of 2. So, whatever the sample size we get using the simple random sampling, we multiply with the 2 and we take the double sample size when we are using a simple random sampling, so that this mistake or this lacuna of contiguity of sample is reduced by taking the design effect of 2, which is double the sample size of the whenever you are using a simple random sampling. So, how do you calculate the sample size? What are the assumptions that are needed about the conditions of a study or to complete a sample size calculation? So, actually this is very difficult and typical for a student or for a researcher how to calculate a sample size. If we really go about the formula and each one separately and what is the definition of each one, if you substitute, that is very easy to understand and to calculate. So, what are needed actually to calculate the sample size? What are the values that are needed? For example, values of estimation of sample size, for example, you choose the main endpoint of interest and the method by which it will be measured. For example, you take difference in average calcium intake from food between two groups using two sample t-tests. How to calculate the sample size for that kind of a scenario? Or else specify the size of the difference between the experimental group that is meaningful to detect. So, actually what happens, we need 3 parameters to calculate the sample size. The amount of the change you are affecting at the end of the study. For example, if you are given 100 iron tablets for a pregnant woman, after 3 months, what is the amount of change in hemoglobin the pregnant woman may get will form you the end result, which you have to substitute in the formula. I am going to tell you the formula and how to substitute that. For example, what is the meaningful difference? For example, in the above calcium intake, meaning difference would be around 225 milligrams. If at all yours is 225 milligrams, what would be the sample size? And the third one is estimate the expected variability. You can ask me how do I get the variation, as I have not yet conducted my experiments. That is why whenever you come to a statistician or any person who knows about calculating the sample size, they ask you what is the variation of your data, what is the amount of change you are going to affect at the end of your study, and what is the confidence interval, and what is the error you are expecting. These 4 things they will ask. Then you can answer saying that, how can I know the variation as I have not yet conducted my study? Then the answer for that is, you have to go for a literature survey or any thesis work, or some people who have done the similar kind of a research and kept as a thesis. You take their standard deviation or variation as if it is yours, and the amount of change to be actually done by yourself or any animal experiment, or your wisdom says that if I give this intervention, this will happen. That is the amount of change, and the variation in the data, and the error. Generally, every time you are, whenever you are referring, that you refer that p is less than 0.05. What does that mean? That we will learn in the subsequent modules of testing of hypothesis, where the error is 5%. That means that if you repeat the experiment 100 times, 5 times you are likely to get the wrong answer, 95 times you get the similar what you are expecting. That's why it is called a 95% confidence interval. So if you see in the picture or in the screen, the formula is given for a sample size calculation for the mean change or mean effect. The formula says, it is a typical formula which says that Z alpha by 2 into sigma, which we call as a standard deviation, the square of that, divided by the epsilon. Epsilon is nothing but the amount of change you are expecting at the end of the study. So these are the 3 parameters we have to substitute. The first parameter, Z alpha by 2, is nothing but the 95% confidence interval or 5% error, which is in general the value is 1.96, which is available in the tables of statistical textbook. You need not do any research for this. This is easily available, and it is already available in the tables given in the statistical textbooks. That is nothing but, if at all you want to have only 5% of error in your study, if you want to conduct the study and you want to calculate sample size, the value for that is 1.96 in the denominator, which we call as a Z alpha by 2. The sigma, which is also called as a standard deviation, where that is not your standard deviation because you have not yet started your experiment. That is a standard deviation you get from any literature search who has already done the research of your nature or similar kind of experiment. Take their standard deviation as if it is yours and substitute. So 2 parameters you already got. The denominator, which we called as epsilon, is nothing but the amount of change you are expecting at the end of the study. For example, if you give the intervention for 3 months or 6 months, what will happen to hemoglobin, what will happen to zinc, or what will happen to calcium, that kind of information if you have. So the amount of change whichever you have at the end of the study, that should be evidence based. We cannot give arbitrarily. Therefore, either you go through any other research already been done by someone else, or you have some information about this, or someone of your colleagues have done a thesis on that and that is already available. Use that and substitute here to get your sample size. For example, I also given, you can see on the screen, I am giving an example how many participants would be needed to have a 95% confidence in mean cholesterol lowering between the high and low fiber diets if the high fiber diet lowers mean cholesterol by 5 milligrams per deciliter more than the low fiber diet with standard deviation of 25 milligrams per deciliter. So it consists of everything. I mean to say 95% confidence interval is nothing but either your experiment is 95% correctly, even you repeat the experiment 100 times, 5% it may go wrong, still it is acceptable. That value is always 1.96, which we are going to substitute now in the formula. And in the example, the standard deviation they are given as 5, where we are substituting there 5 square because that is the sigma square which we are given in the formula. It is not 5, it is 25 standard deviation. The 5 is in the denominator where the amount of change you are affecting at the end of the study. Therefore, now you only have to substitute your value as the amount of change you are expecting at the end of the study, that is the denominator, which you have to pick either from the past research or with your wisdom or someone else already done and kept in the library of the research which you are going to in the similar lines. And the standard deviation is always from the literature search, and that 1.96, which is a 95% confidence interval, is already given in the table. That is a standard mathematical value. If you substitute all these three values, the sample size you got here is 96. So, exactly 96 subjects you can consider for your research. If you don't substitute or use this kind of appropriate formula, if you take 50 subjects, then you are underestimating your results. If you don't use this formula and take 200 subjects, you are wasting resources. Either way, you are doing mistake if at all you don't use the appropriate sample size for calculating your research and for calculating your problem. Therefore, this is the formula. Whenever your data is in terms of mean, to calculate the sample size prior to your research, you have to use this formula. So, every time we are not interested in taking the means only, sometimes we are also interested in considering our research in terms of proportions or percentages. For example, the prevalence of anemia you take, it is 50 or 60% at the present. You want to give some intervention, either food or therapy or whatever way you want, and you want to reduce that 50% to 30%. Therefore, you want to reduce the prevalence of anemia by 20%. How much sample size it is required to go ahead with your kind of a research. The formula which is displayed on the screen or shown on the screen is the same formula, but we use the terms called P and Q. What is P? P is the prevalence of a particular disease. If P is 50% and the normals are again 50%, total is 100% or 100% in the population. Therefore, P and Q are related as P is equal to either 1 minus Q or Q is equal to 1 minus P. If at all your prevalence is 50%, you can also write that as a 0.5 in terms of the proportion. If you see the formula on the screen, the formula says that Z alpha by 2 whole square into P into Q by epsilon. The Z alpha by 2 whole square is always in terms of the error. If at all you are considering 5% error, the value for that is 1.96, which is already available in any of the statistical textbook. If at all your error precision is too small, say 1%, the value is 2.59, you have to substitute. Here the thumb rule is that the more the sample size, the less the variation, the less the error. Therefore, Z alpha by 2 1.96 you can take from the textbook or any of the value, that is a standard. And P, the prevalence of the particular disease or particular symptom, where you consider that as a proportion, 50% means 0.5 proportion, 30% means 0.3 proportion, and correspondingly Q is nothing but 1 minus P, and the epsilon is the amount of change you want to effect in this research. Probably from 50% you want to reduce by 30% means you are going to affect by 20%. Therefore, epsilon will become 20%. Based on that, if you substitute your values into the formula, and you can get whatever the sample size is required for this kind of a problem. Therefore, in this formula which is displayed on the screen or in the picture, you can use whatever the prevalence of interest you can and calculate for different types of prevalences what the sample size you get. That is a homework for you and it is easy to calculate. Only thing is you go ahead with the what are the different sample sizes are required for different types of prevalences, and based on the different type of prevalences and what is the sample size, and based on the different types of the amount of change or the error in the denominator, what are the different sample sizes, you just go on changing the values in the formula and you will get different sample sizes for different types of P's, different types of epsilons, and also if you want to increase the confidence interval from 95 to 99, the sample size also increases. Therefore, the more the sample size, less the variation. So, this is the way you calculate using the prevalences, and previous one we talked about using the means. Today I am going to conclude this session. In this session we dealt with the putting of objective, hypothesis, what are the different study designs used for different situations, where, how and when different study designs are being used, and also different sampling procedures, how to pick a sampling using different sampling procedures. And finally, we used the different sampling size procedures, or how to calculate the sample size when your data is in terms of continuous like means and also in proportions. Therefore, this would definitely enable you how to put an objective hypothesis, how to use different study designs to your different kinds of problems, and how to pick a sample using different sampling procedures, and finally how to calculate the sample size and go for a survey and get the data. So, this finally enables you to get the data and which will be subjected for statistical analysis further.